I'm Professor Kristen Reyher, a veterinary epidemiologist at the University of Bristol's Veterinary School. The farm environment harbors a diverse microbiome made up of billions of bacteria, some of which are helpful and some of which might be disease-causing and harmful to our livestock and to ourselves. Antimicrobial resistance is a major threat to human and animal health worldwide. But to tackle this problem, we first need to understand what drives the selection of resistant bacteria. I'm leading a research team at the University of Bristol as part of Arwain DGC, an ambitious project connecting industry experts, researchers, farmers, and veterinarians to lead the way in responsible antimicrobial use in Wales. As part of this research, my team will be investigating what drives the selection of antimicrobial resistant bacteria on farms. We'll be assessing a range of potential drivers such as use of antimicrobial medicines, certain farm management decisions, and biosecurity. We're following 50 dairy, beef, and sheep farms from across Wales to represent the major livestock farm types we see in Wales. From this diverse sample, we hope to gain an understanding of the levels of resistant bacteria in the on-farm and near-farm environment. To do this, every month our specially trained veterinarians will collect samples from the farm environment. These will be taken from various farm locations, depending on the livestock species and the time of year. For example, we will sample livestock at grass, in collecting yards and milking parlors, as well as in calving and lambing sheds. In previous projects, we have developed and published on an easy and quick method to collect samples directly from these farm environments. The sample collector uses sterile overboot socks worn over wellies to traverse the farm environment, picking up any fecal contamination of the environment on the outside of the sock. The sock can then be removed, sealed, and sent to our microbiology laboratories. Once the sample reaches the lab, we selectively grow the bacteria E. coli from the samples. E. coli is an often harmless bacterium commonly found in the guts of mammals. It is fairly ubiquitous in feces, so we are expecting to find it in most of the samples we take. We then test the resistance of these E. coli to a range of antimicrobials that are often used in livestock, as well as those which are important to human health, such as penicillin, tetracycline, and cephalosporins. Additionally, some of the E. coli samples will be sent for genetic sequencing to identify the genes responsible for antimicrobial resistance. This genetic information could lead to the discovery of new antimicrobial resistance genes, but it will also allow us to start to build up a picture of how resistance may be spreading across farms. After a year of sampling, we'll be able to compose a picture of the prevalence of antimicrobial resistance across Wales through different environmental conditions and during different farm activities, such as lambing time at each time of the year. We'll then analyze this data to look for patterns so that we can start to understand more about which farms have very few or no resistant bacteria and why this might be. We'll share our findings with the farms we sample so that they can also learn more about any antimicrobial resistance present on their farm and potential ways to reduce the threat of antimicrobial resistance in their animals in the future. As well as giving us an accurate picture of antimicrobial resistance in Welsh farming, this research will help us understand how we can prevent its development and spread, as well as safeguarding the usefulness of these important medicines into the future. Alongside the other Arwain DGC project partners, this work will put cutting-edge information about AMR in the hands of Welsh farmers and help make them leaders in responsible use of antimicrobials in farming.